Chapter 12 Mineral Nutrition. The basic needs of all living organisms are essential the same. They require macromolecules such as carbohydrates, proteins, and fat, and water and minerals for their growth and development. This chapter focuses mainly on inorganic plant nutrition, wherein you will study the methods to identify elements essential to growth and development of plants and the criteria for establishing the essentiality. You will also study the role of the essential elements, the major deficiency symptoms, and the mechanism of absorption of these essential elements. The chapter also introduces you briefly to the significance and the mechanism of biological nitrogen fixation. 12.1 Methods to study the mineral requirement of plants. In 1860, Julius von Sachs, a prominent German botanist, demonstrated for the first time that plants could be grown to maturity in the definite nutrient solutions in complete absence of soil. This technique of growing plants in a nutrient solution is known as hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrient essential for plant. The essence of all these methods involve the culture of plant in a soil-free defined mineral solution. These methods require purified water and mineral nutrient salts. Can you explain why this is so essential? After a series of experiments in which the roots of the plant were immersed in nutrient solution and wherein an element was added, removed, or given in varied concentration, a mineral solution suitable for the plant growth was obtained. By this method, essential elements were identified and their deficiency symptoms discovered. Hydroponics has been successfully employed as a technique for the commercial production of vegetables such as tomato, seedless cucumber, and lettuce. It must be emphasized that the nutrient solution must be adequately aerated to obtain the optimum growth. What would happen if solution were poorly aerated? Diagrammatic view of the hydroponic technique is given in figure 12.1 and 12.2. 12.2 Essential mineral elements. Most of the mineral present in soil can enter the plant through roots. In fact, more than 60 elements of the 105 discovered so far are found in different plants. Some plant species accumulate selenium, some other gold, while some other growing near nuclear test sites takes up radioactive strontium. These are techniques that are able to detect the mineral even at very low concentration, 10 to the power 8 gram milliliter. The question is whether all the diverse mineral elements in a plant, for example gold and selenium as mentioned above, are rarely necessary for plant. How do we decide what is essential for plant and what is not? 12.2.1 Criteria for essentiality. The criteria for essentiality of an element are given below. A. The element must be absolutely necessary for supporting normal growth and reproduction. In the absence of the element, the plant do not complete their life cycle or sets the seeds. The requirement of the elements must be specific and not replaceable by another element. In other words, deficiency of any one element cannot be met by supplying some other elements. The element must be directly involved in the metabolism of the plant. Based upon the above criteria, only a few elements have been found to be absolutely essential for plant growth and metabolism. These elements are further divided into two broad categories based on their quantitative requirement. First, macronutrients and second, micronutrients. Macronutrients are generally present in the plant tissue in large amount, in excess of 10 millimole kg of dry matter. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. Of these, carbon, hydrogen, and oxygen are mainly obtained from carbon dioxide and water. while the other are absorbed from the soil as mineral nutrient. Micronutrients or trace elements are needed in very small amount, less than 10 millimole kg of dry matter. These include iron, magnesium, copper, molybdenum, zinc, boron, chlorine, and nickel. In addition to the 17 essential elements named above, there are beneficial elements such as sodium, silicon, cobalt, and selenium. They are required by higher plant. Essential elements can be grouped into four broad categories on the basis of their diverse function. These categories are essential element as component of biomolecules and hence structure elements of cell. Example: carbon, hydrogen, oxygen, and nitrogen. Second, essential elements that are components of energy related chemical compound in plants. Example: magnesium in chlorophyll and phosphorus in ATP. Third, essential elements that activate or inhibit enzyme. For example, magnesium is an activator for both ribulose biphosphate carboxylase oxygenase and phosphoryl pyruvate carboxylase, both of which are critical enzyme in photosynthetic carbon fixation. Zinc is an activator of alcohol dehydrogenase and MO of nitrogenase during nitrogen metabolism. Can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathway you have studied earlier. Fourth, some essential element can alter the osmotic potential of a cell. Potassium plays an important role in the opening and closing of stomata. You may recall the role of mineral as solute in determining the micro water potential of a cell. 12.2.2 Role of macro and micronutrients. Essential element perform several functions. They participate in various metabolic processes in the plant cells such as permeability of cell membrane, maintenance of osmotic concentration of cell soap, electron transport system, buffering action, enzymatic activity, and act as major constituents of macromolecules and coenzyme. Various forms and function of essential nutrients and elements are given below. Nitrogen. This is the essential nutrient element required by plant in the greatest amount. It is absorbed mainly as NO3 minus, though some are also taken up as NO2 minus or NH4 plus. Nitrogen is required by all parts of a plant, particularly the meristematic tissue and the metabolically active cells. Nitrogen is one of the major constituents of protein, nucleic acid, vitamin, and hormones. Phosphorus. Phosphorus is absorbed by the plants from soil in the form of phosphate ions, either as H2PO4 minus or H2PO42 minus. Phosphorus is a constituent of cell membrane, certain protein, all nucleic acid and nucleotides, and is required for the phosphorylation reaction. Potassium. It is absorbed as potassium ion in plant. This is required as more abundant quantity in the meristematic tissue, buds, leaves, and root tips. Potassium helps to maintain an anion-cation balance in cell and is involved in protein synthesis, opening and closing of stomata, activation of enzyme, and in maintenance of the turgidity of cells. Calcium. Plant absorb calcium from the soil in the form of calcium ions. Calcium is required by meristematic and differentiating tissues. During cell division, it is used in the synthesis of cell wall, particularly as calcium pectate in the middle lamella. It is also needed during the formation of mitotic spindle. It accumulates in older leaves. It is involved in the normal functioning of the cell membrane. It activates certain enzymes and play an important role in regulating metabolic activities. Magnesium. It is absorbed by plant in the form of divalent Mg2+. It activates the enzyme of respiration, photosynthesis, and are involved in the synthesis of DNA and RNA. Magnesium is a constituent of the ring structure of chlorophyll and help to maintain the ribosome structure. Sulfur. Plant obtains sulfur in the form of sulfate. Sulfur is present in two amino acids, cysteine and methionine, and the main constituent of several coenzyme vitamins thiamine, biotin, coenzyme A, and ferredoxin. Iron. Plant obtain iron in the form of ferric ions. It is required in large amount in comparison to other micronutrients. It is an important constituent of protein involved in the transfer of electron like ferredoxin and cytochromes. It is reversibly oxidized from Fe2 plus to Fe3 plus during electron transfer. It activates catalyzed enzyme and is essential for the formation of chlorophyll. Magnesium. It is absorbed in the form of manganese ion Mn2 plus. It activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. The best defined function of manganese is in the splitting of water to liberate oxygen during photosynthesis. Zinc. Plant obtains zinc as Zn2 plus ion. It activates various enzymes, especially carboxylase. It is also needed in the synthesis of oxygen. Copper. It is observed as cupric ions. It is essential for the overall metabolism in plant. Like iron, it is associated with the certain enzymes involved in the redox reaction and reversibly oxidized from Cu plus to Cu2 plus. Boron. It is absorbed as Bu3 3 minus or B4O7 2 minus. Boron is required for uptake and utilization of Ca2 plus. Membrane functioning, pollen germination, cell elongation, cell differentiation, and carbohydrate translocation. Molybdenum. Plant obtain it in the form of molybdate ion M. It is component of several enzymes including nitrogenase and nitrate reductase, both of which participate in nitrogen metabolism. Chlorine. It is absorbed in the form of chloride ion, an ion along with Na plus and K plus. It helps in determining the solute concentration and the anion-cation balance in cells. It is essential for water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution. 12.2.3 Deficiency symptoms of essential elements. Whenever the supply of an essential element becomes limited, plant growth is retarded. The concentration of essential element below which plant growth is retarded is termed as critical concentration. The element is said to be deficient when present below the critical concentration. Since each element has one or more specific structure or functional role in the element, in the absence of any particular element, plants show certain morphological changes. These morphological changes are indicative of certain element deficiency and are called deficiency symptoms. The deficiency symptoms vary from element to element and they disappear when the deficiency mineral nutrient is provided to the plant. However, deprivation continues. It may eventually lead to the death of the plant. The part of the plant that show the deficiency symptom also depend on the mobility of the element in the plant. For elements that are actively mobilized within the plant are exported to young developing tissues. The deficiency symptoms tend to appear first in older tissues. For example, the deficiency symptom of nitrogen, potassium, and magnesium are visible first in the ncert leaves. In the older leaves, biomolecules containing these elements are broken down, making these elements available for mobilizing to young leaves. The deficiency symptoms tend to appear first in the young tissue whenever the elements are relatively immobile and are not transported out of mature organ. For example, element like calcium is part of the structure component of the cell and hence is not easily released. This aspect of the mineral nutrition of plant is of a great significance and importance to agriculture and horticulture. The kind of deficiency symptoms shows in plant include chlorosis, necrosis, stunned plant growth, premature fall of leaves and buds, and inhibiting of cell division. Chlorosis is the loss of chlorophyll leading to yellowing in leaves. The symptom is caused by the deficiency of elements N, K, Mg, S, Fe, Mn, Zn, and Mo. Likewise, necrosis or death of tissue, particularly leaf tissue, is due to deficiency of calcium, magnesium, copper, potassium, lack or low level of nitrogen, potassium, sulfur cause an inhibition of cell division. Some elements like nitrogen, sulfur delay flowering in if their concentration in plant is low. You can see from the above that the deficiency of any element can cause multiple symptoms, and that the same symptom may be caused by the deficiency of one of the several different elements. Hence, to identify a deficient element, one has to study all the symptoms developed in all the various parts of the plant and compare them with the available standard tablet. We must also be aware that different plants also respond differently to the deficiency of the same element. 12.2.4 Toxicity of micronutrient. The requirement of micronutrient is always in low amount, while their moderate decrease cause a deficiency symptom and a moderate increase cause toxicity. In other words, there is a narrow range of concentration which the element are optimum. Any mineral ion concentration in tissue that reduce the dry weights of tissue by about 10% is considered toxic. Such critical consideration vary widely among different macronutrients. The toxicity symptoms are difficult to identify. Toxicity level for any element also vary from different plant. Many a time, excess of an element may inhibit the uptake of another element. For example, the prominent symptom of manganese toxicity is the appearance of brown spot surrounded by the chlorotic vein. It is important to know that manganese compete with iron and manganese for uptake and with magnesium for binding with enzyme. Manganese also inhibits calcium translocation in shoe tapex. Therefore, excess of manganese may in fact induce deficiency of iron, magnesium, and calcium. Thus, what appear as symptoms of manganese toxicity may actually be the deficiency symptom of iron, manganese, magnesium, and calcium. Can this knowledge be of some importance to a farmer, a gardener, even for you in your kitchen garden? 12.3 Mechanism of absorption of element. Much of the studies of mechanism of absorption of element by plant has been carried out in isolated cell, tissue, or organ. These studies reveal that the process of absorption can be demarcated into two main phases. In the first phase, an initial rapid uptake of ions into the free space or outer space of cells. The apoplast is passive. In the second phase of uptake, the ions are taken slowly into the inner space. The simplest of these cells, the passive movement of ions into the apoplast usually occur through ion channels. The transmembrane protein that function as selective pores. On the other hand, the entry or exit of the ions and form the simplest require the expenditure of metabolic energy, which is an active process. The movement of ions is usually called flux. The inward movement into the cell is influx and the outer movement efflux. You have read the aspects of mineral uptake and translocation plant in chapter 11. 12.4 Translocation of solutes. Mineral salts are translocated through xylem and along the ascending stream of water, which is pulled up through the plant by transpiration. Pull analysis of xylem sap show the presence of mineral salt in it. Use of radio isotopes of mineral element also substantiate the view that they are transported through the xylem. You have already discussed movement of water in xylem in chapter 11. 12.5 Soil as reservoir of essential element. Majority of the nutrients that are essential for the growth and development of plant become available to the root due to weathering and breakdown of rock. These processes enrich the soil with dissolved ions and inorganic salts. Since they are derived from the rock minerals, their role in plant nutrition is referred to as mineral nutrition. Soil consists of a wide variety of substances. Soil not only supplies mineral but also herbaceous, nitrogen-fixing bacteria, other microbes, holds water, supply air to the root, and acts as matrix that's a stabilizer of the plant. Since deficiency of essential minerals affect the crop yield, there is often a need for supplying them through fertilizer. Both macronutrient and micronutrients form component of fertilizer and applied for as per need. 12.6 Metabolism of nitrogen. 12.6.1 Nitrogen cycle. Apart from carbon, hydrogen, oxygen, nitrogen is the most prevalent element in living organism. Nitrogen is a constituent of amino acid, protein, hormones, chlorophyll, and many of the vitamins. Plant compete with microbes for the limited nitrogen is available in soil. Thus, nitrogen is the limiting nutrient for both natural and agricultural ecosystem. Nitrogen exists as two nitrogen atoms joined by a very strong triple covalent bond. The process of conversion of nitrogen to ammonia is termed as nitrogen fixation. Nature, lightning, and ultraviolet radiation provide enough energy to convert nitrogen to nitrogen oxide. Industrial combustion, forest fire, automobiles, exhaust, and power generating stations are also source of atmospheric nitrogen oxide. Decomposition of organic nitrogen of dead plant and animals into ammonia is called ammonification. Some of the ammonia violates re-enter the atmospheric, but most of the it it is converted into nitrate by soil bacteria in the following steps. Ammonia is the first oxide to nitrile by the bacteria Nitrosomonas and Nitrococcus. The nitrite is further oxidized to nitrate with the help of the bacterium Nitrobacter. These steps are called nitrification. Figure 12.3. These nitrifying bacteria are chemoautotroph. The nitrate thus formed is absorbed by plant and is transported to the leaves. In leaves, it is reduced to form ammonia that finally form the amine group of amino acid. Nitrate present in the soil is also reduced to nitrogen by the process of denitrification. Denitrification is carried by bacteria Pseudomonas and Thiobacillus. 12.6.2 Biological nitrogen fixation. Very few living organisms can utilize the nitrogen in the form of N2 available abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organism is called biological nitrogen fixation. The enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in prokaryotes. Such microbes are called nitrogen fixers. The nitrogen fixing microbe could be free living or symbiotic. Example of free living nitrogen fixing aerobic microbe are Azotobacter and Pagernikia, while Rhodospirillum is anaerobic and Bacillus free living. In addition, a number of cyanobacteria such as Anabana and Nostoc are also free living nitrogen fixers. Symbiotic biological nitrogen fiction. Several types of symbiotic biological nitrogen fixing association are known. The most prominent among them is a legume bacteria relationship. Species of rod-shaped rhizobium has such relation with the root of several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad pea, clover peas, etc. The most common association on roots as nodules. The nodules are small outgrowth in the roots. The microfrankia also produce nitrogen fixing nodule on the root of non-legumes plant. Example, ulnus. Both rhizobium and frankia are free living soil, but as symbiotic can fix the atmospheric nitrogen. Uproot any one plant of a common pulse. Just before flowering, you will see near spherical outgrowth on the roots. These are nodules. If you cut through them, you will notice that the central portion is red and pink, which makes the red nodule pink. This is due to the presence of leguminous hemoglobin or leg hemoglobin. Nodule formation involve a sequence of multiple intrication between rhizobium and roots of the host plant. Principal stages in the nodule formation are summarized as follows. Rhizobium multiply and colonize the surrounding of roots and get attached to epidermal root hair. The root hair cut and the bacteria invade the root hair and infection thread is produced carrying the bacteria into cortex of the root, and then the bacteria are released from the thread into the cell, which leads to differentiation of specialized nitrogen fixing cells. The nodule thus forms stabilizers and direct vascular connection with the host for exchange of nutrients. These events are depicted in figure 12.4. The nodule contains all the necessary biochemical components such as the enzyme nitrogenase and leg hemoglobin. The enzyme nitrogen is a amorphic protein and catalyzes the conversion of atmospheric nitrogen to ammonia. Figure 12.5, the first stable product of the nitrogen fixture. The enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic condition. The nodules have adaptations that ensure that the enzyme is protected from oxygen. To protect these enzymes, the nodule contain an oxygen scavenger called leg hemoglobin. It is interesting to note that these microbes live as aerobic under free living condition where nitrogenase is not operational, but during nitrogen fixing event they become anaerobic, thus protecting the nitrogen is enzyme. You must have noticed in the above reaction that the ammonia synthesis by nitrogenase required is very high. Input of energy 8 ATP for each NH3 produced. The energy required thus is obtained from the respiration of the host cell. Fat of ammonia at physiological pH, the ammonia is protonated to form NH4 ion. While most of the plant can assimilate nitrate as an ammonium ion, the latter is quite toxic to plant and hence cannot accumulate in them. Let us now see how the NH4 is used to synthesize amino acid in plant. These are two main ways in which this can take place. First, reductive amination. In this process, ammonia reacts with alpha ketogluaric acid and forms glutamic acid as indicated in the equation given below. Second, transamination. It involves the transfer of amino group from one amino acid to keto group of a keto acid. Glutamic acid is the main amino acid from which the transfer of NH to the amino group take place, and other amino acids are formed through transamination. The enzyme transaminase catalyzes all such reactions. For example, the two most important amides, asparagine and glutamic, found in the plant are the structured part of protein. They are formed from two amino acids, namely aspartic and the glutamic acid, respectively, by addition of another amino group to each. The hydroxyl part of the acid is replaced by another NH2 radical. Since amides contain more nitrogen than the amino acid, they are transferred to other parts of the plant via xylem vessel. In addition, along with the transpiration stream, the nodule of the same plant, example soybean, export to fixed nitrogen as urates. These compounds also have a particularly high nitrogen to carbon ratio. Summary: plant obtain their inorganic nutrient from air, water and soil. Plant absorb a wide variety of mineral elements. Not all the mineral elements they absorb are required by plant. Out of more than 105 elements discovered so far, less than 21 are essential and beneficial for normal plant growth and development. The elements required in large quantities are called macronutrients, while those required in less quantity are traced as termed as micronutrients. These elements are either essential constituent of protein, carbohydrate, fats, nucleic acid, etc., and or take part in various metabolic processes. Deficiency of each of these essential elements may lead to symptoms called deficiency syndrome. Sclerosis, necrosis, stunted growth, impaired of cell division, etc. are some prominent deficiency syndrome. Plant absorb mineral through roots by either passive or active process. They are carried to all part of the organism through xylem and along with the water transport. Nitrogen is very essential for the substance of life. Plant cannot use atmospheric nitrogen directly, but some of the plant is association with nitrogen fixing bacteria, especially roots of legumes can fix the nitrogenic bacteria into the biologically usable form. Nitrogen fixation requires a strong reducing agent and energy in the form of ATP. Nitrogen fixation is accomplished with the help of nitrogen fixing microbes, mainly rhizobium, the enzyme nitrogenase, which plays an important role in biological nitrogen fixation, is very sensitive to oxygen. Most of the process take place in anaerobic environment. The energy ATP required is provided by the respiration of the host cell. Ammonia produced following nitrogen fixer is incorporated into amino acid as the amino group.